Thank you everyone for coming to the talk, and thank you to the organizers for giving me an opportunity to speak. The work that I'm going to be presenting is the result of collaboration with several different people all listed here. And in particular, I'd like to highlight Lucy Stewart, Shrishti Kashyap, and Begum Tapshuolu, who were former and current PhD students in my lab who did this work, as well as some of my co-PIs, Julie Huber from Woods Hole Oceanographic, Darby Dyer from Mount Holyoke College, and Susan Lang from the University of South Carolina. And the work was funded by the Gordon and Betty Moore Foundation and by NASA. And a lot of the work that I'm going to be showing was published just within the last year. And if you're interested in it, it's cited in the AbSciCon abstract. All right, so we have a rover on Mars, and we have ROVs and AUVs in the deep ocean that are searching for life and trying to figure out what that life is doing. And soon we'll have robots and ocean worlds in our solar system. And it's not a big, I don't need to work hard to convince you all that we need both modeling and detection in order to be able to determine where that life is and what that life is doing. But what I've been asking myself, and what I wanted to talk about in this presentation, is how can we better integrate these two? What can I do in my lab, and what can we do as a community to try and bring these two things closer together? And so that's what I wanted to talk about. So first of all, in the area of modeling, I've been working with Lucy Stewart, Chris Alger, Caroline Fortunato, Julie Huber, and others to develop a Methanogenesis reactive transport model. And the questions that we're trying to determine is, for an individual event, how many methanogens are needed to be at that event to create, say, the methane anomalies that we see? What's the volume of rock that's occupied by these methanogens? How deep is the biosphere? And what's the residence time of the fluid? So what we've done is we've developed a one-dimensional pipe flow reactive transport model with a series of boxes, 350-degree hydrothermal fluid flowing through the boxes, and each box you have a little bit of dilution of seawater, and then once the temperature permits, methanogens can then grow in the box before the fluid flows to the next one. We also can vary the size of the box so that we can vary the residence time of the fluid through the box. The model is informed by chemostat work that was done in my lab using both a thermophilic and a hyperthermophilic methanogen, and we determined hydrogen monode kinetics for methane production and Arrhenius kinetics for temperature effects for each of these organisms to be able to plug into this model. What we found is that if the pipe is straight, in other words, if residence time remains more or less constant with fluid flow, is that the hyperthermophilic methanogens dominate the system. They get at the hydrogen first, and they consume it all before the thermophiles really have an opportunity to be able to use that. However, if the pipe is more flanged and residence time increases with flow rate, or with flow, then what will happen is that this gives an opportunity for the thermophiles to be able to dominate the system over hyperthermophiles. So we wanted to actually test this model, and we went to axial seamount off the coast of Oregon and looked at two different hydrothermal vents. And the first two columns, you can see hydrogen and methane data that was collected by Dave Butterfield. And in this column here, you can see cell count estimates for the two different methanogens that's based on metagenomic work that Julie Huber's lab did, and fit the model to it. The black line represents the model, these circles represent the data of what we actually measured. And in these figures, imagine that the 350-degree fluid is rising up and is getting diluted. This is magnesium concentration here on the side. But the fluid is rising up, cooling and mixing as you go up towards the top. And the model did a pretty good job. You can see hydrogen consumption here, methane production here. And in this particular system, marker 33, there are more hyperthermophilic methanogens, so that suggests that the pipe flow at the particular vent is more straight, that residence time into each box is more or less constant. Whereas at marker 113, there are far more thermophilic methanogens than hyperthermophilic methanogens, and that again suggests that perhaps as the fluid flows, the residence time at each temperature gets longer and longer. Some of the things we're able to determine from this is that there's only about 10 to the 11th methanogens that are necessary at each vent to be able to create the methane anomaly that we saw. That was a surprise. We thought that was going to be much higher than it really was. The residence time at methanogen growth temperatures was about 29 to 33 hours. Only about 2 to 18 cubic meters of seafloor was occupied, depending upon the porosity of the rock. And the biosphere is, we think, is only about 2 to 30 meters deep. Moving forward, we need to put more metabolic diversity into these types of reactive transport models. This is something that we're working on now. And also, we need to put more ecological theory into these models. We're also, in terms of the actual detection part of it, we looked at methane fractionation. So I work with Begum Tapshuolu, Tran Nguyen, Susan Lang, and others to look at the methane carbon fractionation that occurs in a pure hyperthermophilic methanogen. And we varied the flux rate again in a chemostat to see how the organism grew, how gene expression changed, and how carbon fractionation changed. When hydrogen flux was high, what would happen is the carbon would flow through the Wood-Ljungdahl pathway, and you'd see more methane and more energy produced. And in that particular situation, carbon fractionation was relatively low, about an epsilon of about 29 part per thousand. However, when hydrogen flux rate was low, the carbon would actually flow more into biosynthesis, so there'd be a much higher cell yield. And also, carbon fractionation increased significantly. The epsilon would go to about 70 to 85 part per thousand. And axial seamount, which I just told you about, the carbon fractionation we think is pretty low, so that actually fits the model that we think that there's probably a high flux of hydrogen that's feeding the methanogens in that system. Moving forward, we need to do more in terms of understanding carbon use efficiency for a variety of different organisms, how that might vary with change in nutrient availability. And also, we need to work on more biomarker information. What does carbon fractionation look like in, say, lipids and proteins? So also in the area of detection, moving beyond methanogens, we're interested in what happens with minerals. Could minerals provide, say, a different type of biosignature? So I work with Shrishti Kashyap, Eli Skloot, and Darby Dyer and others to synthesize 6 different nanophase iron oxides that you see listed here. And we work with 2 hyperthermophilic iron reducers, and Pyridictium delaney and Pyrobaculum icelandicum, that grow on iron oxide. They reduce the iron oxides. And what we found was that the organisms grow best on ferrohydrite. They grow modestly well on lepidocrosite and accoganiite, and they grow rather poorly in maghemite, goethite, and hematite. And that pretty much fits what we would expect based on the increasing order of crystallinity and the thermodynamic stability of the minerals themselves. And I'd like to point out, too, that my PhD student, Shrishti Kashyap, will be speaking in more detail about this particular project on Friday. And she's soon to be on the postdoc market, so she's very good. So what we then wanted to do is we wanted to try and identify what those minerals are that were formed by these iron reducers. And remember, when the organisms were growing on ferrohydrate, they grew best. They had the highest Fe2 flux rates, production rates. And what we found was that we used, by the way, a combination of VNIR, FTIR, Raman, and Mossbauer. They all pretty much gave us the same results, but the FTIR data is shown here. But when the organisms were growing on ferrohydrite, they produced magnetite was their end product. However, when the organisms were growing on lepidocrosite and accoganiite, remember this is slower growth rate, slower Fe2 production rate, the mineral end product actually changed. With lepidocrosite, they produced an iron carbonate, a siderite or a siderite-like compound mineral. And when they were growing on accoganiite, the end products were vivianite, which is an iron phosphate, as well as some magnetite. And it's a little hard to tease out in this picture, but I certainly would be happy to talk about it in more detail if anybody's interested in this. But we did see some signals that suggest that perhaps there is more, in some cases, a biogenic type mineral transformation product compared to just abiotically synthesized minerals. But that's something we still are working on and need to tease out more. So moving forward into the future, and especially in the area of detection and also in the areas of how can we couple modeling and detection together, We're now in the process of taking pure thermophiles and hyperthermophiles and getting spectra from them using these 4 different spectroscopy techniques. This is FTIR here. And these are all metabolically very different thermophiles and hyperthermophiles. And we do see differences, especially in the near IR and mid IR range. We do see some differences between these different organisms. So that's very tantalizing for us. And we're hoping we can get to the point that we might actually be able to use spectroscopy in a native rock or even on the seafloor to be able to see what types of organisms are there. But that's still a dream, so still a ways off. We're also interested in hyperspectral imaging. This is a piece of sulfide that was sitting on my shelf in my office for 10 years, and we decided to put it under a hyperspectral image camera, and it's about 3 centimeters across. This is just the RGB image here, but what you can do is you can do a pixel by pixel analysis looking at the spectral information for each pixel and get spectra for each pixel and then map out where you see similar spectra. And this actually did a fairly decent job of mapping out where the different minerals were in this. Long term, what we really hope we would be able to do is to be able to do something like hyperspectral imaging and to be able to co-locate different mineral types and different microbes, or at least some sort of a biogenic signal. And even maybe be able to do this not only on fresh collected samples from hydrothermal vents on board ship, but maybe even one day on the seafloor itself. So the take-home message from all this is that we really need to improve the iterative process of working between modeling and detection. And these are fairly obvious reasons, but it's worth stating is in order for us to really be able to predict where to find life and what kinds of life we would find, also determining the impact of that life, and really to be able to ground truth the modeling results that we're generating. So thank you very much, and I'm happy to take any questions.
Have time for questions.
Hi, Aaron Noel from JPL. Really nice talk.
Thank you.
Did you all ever do any sort of chemical analysis besides genomic work? Do you look for other biomarkers?
So we have done a little bit of looking at the methane and the carbon fractionation that's there. And we haven't done any lipid analysis or protein in terms of like extracting that and looking directly at that. But that's something that we hope to get into doing more, especially if we can do some analysis on the carbon fractionation that actually occurs in the lipids or at the protein level. So that's one of the areas that we're trying to move into in the future.
Great, thanks.
More questions? Just come to the microphone.
Hi, John. How's it going? Thank you for the talk. I was wondering, in your pure culture experiments with those mineral phases, did you notice any variation in the mineral end product if you changed up the electron donor?
We haven't tried that, but it's a great question. Especially for the pyrobaculum species, it's a facultative autotroph. So we've grown it with organics, but we'd love to do it autotrophically. The pyrodictium species uses hydrogen and the pyrobaculum uses organics, and we do see some differences there between the two. We don't know if that's at the organismal level or how much of it is because they're using a different electron donor source. But Jill, that's a great question.
Hi, great talk. Varun Paul from Mississippi State. I was curious about the flange pipe versus the straight pipe. Was there a difference in the temperature in the straight pipe versus the flange pipe that could affect thermophiles being dominant in the flange pipe because it broadens out, the temperature drops? Were you able to measure the temperature difference?
Well, you can see in the modeling, in some ways each step you might expect that there's the temperature inside the box the same, but that's really the residence time inside that box that really has a big impact. So for example, if hyperthermophiles spend very little time in their box before you move on to the thermophile temperatures, then the thermophiles have a chance to dominate the system. But if hyperthermophiles get to the hydrogen first and the residence time is long enough, then they'll dominate the system.
Thank you.
Hi, real quick question. I was wondering what the flow rates you were considering of in terms of the residence time in some of your chemostat experiments and how that would be affected by, say, a diffusely flowing system like Lost City, for example, where you have very low flow rates and really high thermal diffusion through the system?
So we varied the hydrogen concentration or the hydrogen flow rate into the reactor, and of course, as we did that, that changed growth temperature, the growth rate, and we had to vary the dilution rate to match that. We did try a couple different dilution rates for our chemostats at any given hydrogen concentration to see how much dilution affected the system, and we found that at least statistically there wasn't really that much difference with change in dilution rate. It really seemed to be primarily driven by the hydrogen concentration, but that is something that you have to integrate into chemostat experiments, since growth rate varies as the hydrogen flux varies.
Great. Let's give one more round of applause to Jim Holden. And those of you in the back, there's a lot of room up.